Lecture number 3. Pathogenic vibrios, causative agent of cholera, Helicobacter pylori, and Campilobacter. Cholera. Cholera-like disease were known from the times of ancientity. In 1817, first cholera pandemic occurs in Asia, and Robert Koch was the first who discovered causative agent of cholera. And there were 7 pandemics of cholera in the world, and nowadays we also have incidence of cholera in 20 in 2010 year, it was a great epidemic of cholera in Haiti, and it was caused by Helocobabacter. And several countries in Africa and Asia have reported cholera outbreaks. Major ongoing outbreaks are being reported from Bangladesh, Nigeria, and Niger. In 2021, new cholera cases have been reported worldwide, and countries reporting the majority of new cases since the previous update are Nigeria, Niger, India, and Bangladesh. So you should understand that cholera is not ancient disease, and we can meet cholera in our day, and especially near the countries of Africa and Asia. Classification. Causative agent of cholera belongs to order Vibrionaris, family Vibrionacea, genus Vibrio, species Vibrio cholera. There are more than 200 serogroups of Vibrio cholera, and it is because of O antigen of cell wall, and cholera is caused by serogroup O first and O 139. Classical Vibrio cholera biotype and L-Tor Vibrio biotype pertain to O1 serogroup. Representatives of Vibrio cholera beyond the serogroups O1 and O139 are accidental agents of moderate human diarrhea disorders being of lesser clinical relevance. Another members of Vibrio genus, like Vibrio parahemolyticus, may cause diarrhea in humans. In addition, Vibrio vulnificus, it's a causative agent of human wounds infection. But it is not so spread and not so important as cholera. Vibrio cholera is common-shaped gram-negative curved rods, non-sporoforming. It can has a capsule, and also it has very strong polar flagellum. Possess numerous pili responsible for microbial colonization. Curved rods with polar flagellum and capsule. Pili for adherence, and hemagglutinin for adherence also. Vibrio cholera is aerobic or facultatively anaerobic bacteria. Actively grow on basic nutrient media. The temperature range for culture is from 14 to 42 degrees. Vibrios can withstand alkaline pH, and it resistant to bile salt. On solid nutrient media, Vibrio cholera produce opaque, granular, smooth, round, and convex dome-shaped colonies with a light blue shine. In alkaline peptone, Vibrio cultures form a pellicle that contains agglomerated cholera vibrios. Various enrichment and selective media, for example, alkaline MPA or alkaline nutrient browse, are applied for cultivation, Vibrio cholera, and also thiosulfate citrate bile sucrose can be applied for cultivation, and Vibrio cholera produce yellow colonies on TCBS egg. Here you can see TCBS agar, and the yellow colonies is formed by Vibrio cholera, and green colonies is formed by non-cholera vibrions like Vibrion parahemolyticus. Biochemical properties. Vibrios are oxidase positive, and they ferment various substances with acid and products. Heiberg divided vibrios into 8 groups according to their biochemical activity. Both classical Vibrio cholera biotype and Eldor Vibrio pertain to one Heiberg's group and ferment sucrose and mannose, whereas ribinose and lactose not. Bacteria produce ammonia, indole, and reduce nitrates to nitrates. Also, they secrete a number of proteins, coagulate serum and milk, liquefy gelatin, cholera vibrios render variable hemolytic and hemagglutinating properties. Antigenic structure. Vibrio cholera has heat label H antigen of flagella and also heat stable O antigen. By O antigen, Vibrio cholera divided into 200 serogroups, but medical significance has only 2 serogroups. It's a serogroup O1 and serogroup O139. It was mentioned above that classical Vibrio cholera biotype and Vibrio cholera biotype L2 pertain to O1 serogroup. O1 antigen contains A, B, and C antigen variations. Thus, remain serotypes with O1 serogroup were established: Ogawa, Inaba, and Kikushima. Vibrio cholera O139 was proven to originate from L. Vibrio. It happened after acquisition of gene cluster encoding the synthesis of novel O139 L-base antigen, which thereby substituted initial O1 antigen of L. Here, virulence factor. Vibrio cholera has common virulence factor like adherence by pili, lipopolysaccharide, endotoxin, exopolysaccharide, which can help biofilm formation, capsule, and enzyme hemagglutinin protease, by which Vibrio cholera can detachment from enterocytes, thereby promoting further microbial spread along the intestinal wall. But major valence factor is enterotoxin choleragen. It composed of 2 subunits, A and B, and 1A and 5B. B subunits, it's like transport subunits. It binds to the intestinal cell via cell membrane ganglioside receptor. And subunit A is translocated through cytoplasmic membrane into intestinal epithelial cell, undergoes TOL-dependent activation, and promotes ADP ribosylation of cell G proteins. This stimulates cellular adenylate cyclase, resulting in great increase of C and P concentration. The rise of intracellular C and P concentration blocks active sodium chloride absorption and increased chloride and bicarbonate secretion. The later result in passive water loss with development of massive diarrhea. This is followed by marked decrease of intravascular volume, life-threatening hypoperfusion of critical organs and hypotension. Okay, cholera toxin, it consists of 5 B subunits and 1 A subunits. B subunits bind to receptors on the surface of enterocytes and help A enter to the cell. Subunit A cause ADP rivalization and make increasing the level of CMP. And because of increasing secretion of CMP, cell began to secrete to lose electrolytes and water, and that's why diarrhea and dehydration occurs because of losing of this water of the cell by the cell. Resistance. Naturally, vibrions live in water, and in history we have some information that before first cholera pandemic, Vibrio cholera was usual water microbes which lived in water and don't cause any infection. But after changing of condition of ecological condition, Vibrio cholera start to mutate and get a possibility to produce cholera toxin, and start to become pathogenic. So Vibrios can live in aquatic ecosystem, and they colonizate zooplankton, plants, and algae, and have possibility to live a long time in water, 1 month in sea and river, and 2 days in various foodstuff. In feces, about 1 month. It readily survive at low temperature, but sensitive to heating, ultraviolets, and desiccation, and very susceptible for disinfectant treatment. Low concentration of hydrochloric acid inactivate bacteria within 1 minute. Pathogenesis. Cholera vibrios inhabit the water or rivers, seas, and oceans. Most environmental O1 strains are lack of choleragen expression, but only toxigenic Vibrio cholera can cause the disease. It is very important that non-toxigenic vibrions, they can't cause the disease, and non-toxigenic bacteria can get gene of choleragen producing by gene exchanging process, especially in the process of transduction with several temperate bacteriophages. Cholera is anthropootic disease. It is transmitted from sick person and carriers by fecal oral route with infected food or water. The causative agent is also carried by flies or can be transmitted by contact route through contaminated hands. Short incubation period of disease lasts from several hours to 5-6 days. After oral injection of of contaminated water or food, most of Vibrio and cholera are killed by acidity of gastric juice. Thus, the infection dose for cholera vibrios is rather high. The rest of bacteria colonize the intestinal epithelial cell of small intestine attached to microvilli and ultimately start to produce very strong, very high enterotoxin choleragen. Action of cholera enterotoxin leads to the development of disease symptoms. So, all pathogenesis is because of choleragen. Choleragen influence on the enterocytes, and enterocytes start to lose its water, and water diarrhea began, and stool and feces lose its features because because it is very strong diarrhea, and all nutrients, all water is leaving organism by process of diarrhea, and that's why fat cells not look like as fat cells, and patients do resemble rice water and contain many epithelial cell, mucus, and large number of fibrils. In several cases, profuse watery diarrhea and continuous vomit results in lowering of body temperature, and it is very important, hypovolemic and hypotensive shock, with lethal outcome within first 12 hours of disease. The total fluid loss can achieve 20-30 liters per day in adults. Without adequate compensatory infusion therapy, the mortality rate exceeds 20%. Abortive and mild disease without any symptoms are observed in majority of cases of L. or Vibrio cholera. Patients' carrier state rarely exceeds 1 month. Here you can see how cholera entered to the human body by fecal oral route, and then main barrier is gastric juice, and if it is enough cholera vibrions and acids is low and level of acids is lower than normal, Vibrio cholera goes to the intestine and uh, attached to the epitheliocytes and destroys the microvilli and cause some inflammation. But the main in pathogenesis is production of cholera toxin, which enter to the cell and increase adenylate cyclase activity, cause the increasing of the level of CMP, and the CMP cause losing of electrolytes and water by cell, and that's why diarrhea occurs. And by diarrhea, human organism lose a lot of water, and dehydration and shock occurs then. Post-infection immunity is high grade but of short duration. The immunity is both antibacterial and antitoxic. Antitoxic antibodies confer most efficient protection against the disease. Laboratory diagnosis of cholera. Specimen, stool, vomit, autopsy, material, water, foodstuff. Methods of rapid diagnosis is dark field microscopy and immune fluorescence assay. By immune fluorescence can be observed feces which contain a lot of vibrions, and feces is By feces, from feces is getting glasses, and then it observed with specific cholera serum. So cholera serum contain antibodies against Vibrio cholera antigen, and these antibodies are marked with fluorochrome. If in the fetum are vibrions, they interact with anti-cholera antibodies and start to shine, start to bright in the microscope. Cultivation must be done only in specific laboratories which have possibility to work with dangerous pathogen, which are locked with closed system of ventilation, closed system of canalization, and this laboratory must be must have all system to prevent spreading infection outside the laboratory. The specimens are inoculated into alkaline peptone water to get a biofilm. Then biofilm must be observed by microscopy with gram staining, and also by slide agglutination with O1 and O139 antiserum. In the first alkaline browse cultivation result, if the first alkaline browse cultivation result in scarcel microbial growth, the material is inoculated again into alkaline peptone browse. After primary examination, alkaline browse culture is planted onto alkaline agar or TCBS. On TCBS, Vibrio cholera grows with yellow colonies, and then yellow colonies examined by slide agglutination with anti-cholera serum, and Vibrios must give a positive result with anti-cholera serum. Then obtain pure culture and identified by morphological, biochemical, cultural features, and of course by antigenic features. Identification by antigenic features must be made by agglutination reaction with O1 and O139 serum. And by this reaction, zero group will be established. Here you can see morphology and the results of gram staining, gram negative curved rows. And here you can see TCBS agar, yellow colonies, and also on the blood agar, vibrions form colonies with hemolysis. Oxidase test grows in liquid media like biofilm. And very important also are molecular genetic methods, and they are used in specialized reference centers for epidemiological studies. Treatment. The organ treatment of cholera is based mainly on infusion, replacement therapy, and compensators the loss of water and electrolytes. Different antibiotics affecting gram-negative flora can be used to facilitate convalescence. Usually oral tetracyclines and are administered. Antimicrobials, chemoprophylaxis, and vaccine prophylaxis may be used for disease prevention in family contact person. For specific prophylaxis, Phenol-killed vaccine and choleragen toxoid are occasionally used now. Nevertheless, they confer only the short-term protection for 6 or 12 months in 50, 80 of vaccinated individuals. So it is not very effective. Non-specific prophylaxis includes all measures to prevent spreading the infection, to prevent infection by from water, from foodstuff, and in the case of cholera, it is necessary to make all measures to prevent spreading infection, lockdown for patient, and of course lockdown for all people who contact with him, and all measure all measure for disinfection. Next pathogen is Helicobacter pylori. Um, in 1982, the Australian physician Barry Marshall and pathologist Robin Warren isolated spiral bacteria from gastric tissue biopsy of patients with chronic gastritis. By the experiment of self-infection, Marshall and colleague proved for the first time the association between this bacteria and the development of chronic gastritis. In 1989, the novel pathogen acquired its final taxonomic name, Helicobacter pylori. And in 2005, Marshall and Warren were awarded Nobel Prize in Physiology or Medicine of their outstanding discovery. Now we exactly know that Helicobacter pylori is associated with chronic gastritis and ulcer disease. Classification. Causative agent is belong to the family Helicobacteriaceae, genus Helicobacter, and there are about 39 microbial species: Helicobacter pylori, Helicobacter hellmanni, Helicobacter mustele, Helicobacter pales, and many others. But the main agent of human disease is Helicobacter pylori. Morphology. Helicobacter pylori is a short or medium-sized gram-negative bacterium of S-like spiral-shaped microbial cell. It has flagella attached to one pole of bacterial body. They have no spore or capsule. Here you can see electronic photo and light microscopy photo. Cultivation. Helicobacter pylori is highly fastidious agent, propagating only in microaerophilic and capnophilic gaseous conditions. So it is too difficult to cultivate Helicobacter pylori. In standard aerobic and anaerobic surroundings, the bacteria can't grow. Else, that's why it was so a long time to discover this bacteria. Also, they have a narrow temperature optimum for growth near 37, being completely inactivated in 25, 28, or about 41 degree. This is not 400, but 41. Helicobacter pylori requires special and selective nutrient media with multiply growth factor. It can be cultured in blood or serum agar, supplement with broad spectrum antimicrobials that inhibit the propagation of contaminant bacteria. Primary growth is evaluated in 5-7 days of culture. So cultivation is very hard because Helicobacter pylori is very fastidious bacteria. The chemical properties oxidize and catalyze positive, express multiplies enzymes like phosphatase, phospholipase, hyaluronidase protease, produce hydrogenic sulfate, demonstrate remarkable urease activity, utilize amino acids as nutrients from available carbohydrates, they metabolize only glucose. Antigenic structure: O antigen, H antigen, and superficial outer membrane proteins, O and B, which are type specific. Virulence factor: Helicobacter pylori produce a number of adhesins, aggressive enzymes, and toxins. The major role in pathogenesis of Helicobacter infection belongs to microbial exotoxin, cytotoxin SIGA, cytotoxin associated gene A, and vacuolating cytotoxin VEC-A. Cytotoxin A is present in most of the virulent strains of Helicobacter pylori. The main pathogenic function of cytotoxin includes the impairment of cellular metabolism and activation of cell-mediated inflammatory reaction. Vacuolating cytotoxin binds to membranes of gastric epithelial cell. It demonstrates pleiotropic pathological effects against gastric mucosal membrane. For instance, this toxin provokes the secretion of pro-inflammatory cytokines by leukocytes. Moreover, the molecule of toxin creates membrane pores, allowing their own entry into epithelial cell. When entered into the cell, the molecules of VEC-A toxin trigger cell apoptosis, or at least they cause profound degenerative changes in gastric mucosa, cell vacuolization, and disruption of cell-tight junction. So, inflammation and cell apoptosis, it's main parts in pathogenesis of Helicobacter infection. Helicobacter pylori intensively produce a number of aggregation and invasion enzymes. Microbial urease catalyze urea decay. They lead to the production of exuberant amounts of ammonia that not only damage the mucous tissue but also neutralize the acidity of gastric juice, thus fostering microbial survival. When we talk about when we talk about gastric juice, you should know exactly that all bacteria are sensitive for gastric juice action, but Helicobacter pylori has possibility to destroy this barrier by microbial urease, and it gives them possibility to survive in gastrointestinal tract and to cause all damage that it can make in this biotope. So, by microbial urease, Helicobacter pylori can survive in the stomach. Also, hyaluronidase and microbial flagella stimulate bacterial invasion into submucous gastric layer. Microbial phospholipase destroys the membranes of epithelial cell. Here you can see all virulence factors: flagella to motility and to invasion, urease that neutralize gastric acid and give possibility to survive in the stomach and in the duodenum. Lipopolysaccharides adhere to host cell inflammation, and exotoxins, there are two exotoxins which destroy cell, cause the apoptosis of the cell, and also cause inflammation. And that's why gastric mucosal injury occurs. and also secretory enzymes like mucinase, protease, lipase, and toxin which leads to uh, production a lot of inflammation mediators, a lot of cytokines, and that's why inflammation occurs. Generally, helicobacters are low resistant bacteria, taking into account the narrow temperature range, 34-40 of their growth and toxic action of atmospheric oxygen. Nevertheless, there are some individual reports about Helicobacter survival in dental plaques, saliva, vomits, and gastric juice. Pathogenesis. The source of Helicobacter infection is infected humans, so anthropootic infection. The routes of transmission are not completely established. In most cases, the infection is transmitted by fecal-oral mechanism or by direct contact. Iatrogenic spread of infection via contaminated endoscopic equipment also can be excluded. When entered the stomach, most of the bacteria settle in gastric antrum, where the local pH of mucosal tissue is higher. Next, they move towards duodenum. Active locomotion of microbial cell promotes their invasion into a submucous gastric layer. Here they attach to membrane glycolipid receptors. Urease of Helicobacter pylori metabolize urea with ammonia release that neutralize the acidity of gastric juice, supports long-time microbial survival, and directly damages gastric mucosa. The most virulent are Helicobacter strains with parallel production of both bacterial cytotoxins, CIGA and BACA. CIGA interference in normal life cycle of epithelial cells. Pteroglycan fragment stimulate inflammatory response via activation of transcription factor NF. Together with BAC-A toxins, they promote the development of acute gastritis and or duodenitis. So exotoxins, they cause inflammation and cell destruction. One toxin, WAC-A, is responsible for cell destruction because it causes apoptosis. And the CAGA toxin, it causes inflammation by stimulation stimulation cytokines secretion. This is followed by local hyperproduction of pro-inflammatory cytokines, interleukin 8 and others that stimulates neutrophily and lymphocytes infiltration in stomach wall. Diagnosis. Two groups of laboratory methods are used for detection of Helicobacter pylori, non-invasive and invasive tests. The latter needs gastric biopsy specimen. Rapid non-invasive carbon urea breath test discovers urease activity of Helicobacter pylori. The test is convenient for mass screening of people attending medical offices and clinics. When tested, the examined person drinks urea solution, radioactivity labeled with under the action of microbial urease labeled uh, uh, carbon oxygen is released that is registered in expired air. Other non-invasive tests include determination of antigens of Helicobacter pylori infections by ELISA test and detection of microbial DNA by PCR. Here you can see non-invasive carbon urea breath test. Patient must drink specific urea solution. which contain labeled uh, labeled carbon hydrate, and and then it can be established by the changing um, of the uh, specific um, reagent. Invasive test presumes the examination of gastric biopsy specimen taken during endoscopy. For instance, rapid urease test detects the Helicobacter urease in gastric biopsy by placement of specimen into urea solution. The decay of urea is followed by ammonia accumulation that elevate pH of the medium and changing the color indicator dye. The most reliable test for direct detection of Helicobacter pylori is biopsy specimen is microscopy with histological hematoxylin eosin staining or Wharton stairs silver strain that is more sensitive. For isolation of microbial culture, the tissue specimen is inoculated into special media supplement with antibiotics and multiply growth factors. Incubation is performed in microaerophilic condition with increased concentration of carbon oxygen. Uh, Primary growth should be assessed in 7 days. The isolated culture is further examined by microscopy, biochemical testing, serological and molecular genetic tests. But as I said previously, cultivating of Helicobacter pylori is not easy uh, because of um, requirement of this microbe. It is very fastidious microbe, and this method can be applied in the case of uh, antibiotic resistance to know to which antibiotics this microorganism is sensitive only by this reason, because it is too hard to find to get pure culture of Helicobacter pylori. To diagnose this, it is more easy detect Helicobacter pylori by PCR or by ELISA than by pure culture method. Serological diagnosis use ELISA test for evaluation of specific antibodies in patient serum directed against Helicobacter pylori antigens. Treatment to prevent unfavorable consequence of Helicobacter pylori infection, complete eradication of this pathogen is required. Recommended first-line antimicrobial treatment, so-called triple therapy, includes proton pump inhibitors, omeprazole, and two antibiotics, amoxicillin and clarithromycin. Microbiological confirmation of eradication is performed after the end of treatment course. In case of first-line treatment failure, quadruple therapy is used expanded with colloidal bismuth salts in the light of growing antimicrobial resistance of Helicobacter pylori. Antibiotic of other groups can be administered like metronidazole, tetracycline, and fluoroquinolones. Despite the high frequency of successful eradication, the cases of reinfection with Helicobacter pylori are common mainly due to the broad spread of this agent among the human population. Prophylaxis is non-specific. There is no any vaccine, and it is based on general measure for efficient sterilization of medical instruments, antisepsis, and disinfection. Okay. And the last pathogen is Campilobacter, and It is microbial agent which responsible for diarrhea, abdominal pain, spasm, blood in the facies, nausea, vomiting, and fever. History of discoveries. The first description of Campilobacter was made in 1886 by Theodore Escherich, and he observed and described bacteria which cause enteric disease very similar to cholera infantum. And in 1906, John McFadden and Stewart Stockman, they report the presence of large numbers of peculiar organisms in loafers' blue stained smears of uterine mucosa of pregnant Eve. And Campylobacter jejuni is a leading cause of enteritis and enterocolitis worldwide. Also, Theodor Escherich himself provided drawing of Helicobacter back in 1880s. Campylobacter jejunus was not identified until 1950. Classification. Order Campylobacteriaceae, family order Campylobacteriales, family Campylobacteriaceae, genus Campylobacter, and there are a few species, but the main are Campylobacter jejuni and Campylobacter fetus. Campylobacter jejuni is causative agent of gastroenteritis, extraintestinal infection, Guillain-Barré syndrome, reactive arthritis. And Campylobacter fetus is causative agent of vascular infection and meningoencephalitis and gastroenteritis. Morphology. The word Campylobacter came from a Greek word which means carved road. It is also gram-negative, spiral S-shaped bacilli, non-sporing, and with flagella and capsular polysaccharides. Here you can see carved rows, and it has flagella. And sometimes in some literature we have we can find the word wings-like bacteria, because of its shape. Cultivation. As it was in the case of Helicobacter pylori, cultivation is not easy because of requirement of this microorganism. Campylobacter jejunum is thermophilic and grows better at 42 degrees, but other species are mesophilic and require 39 degrees. And also they grow best in an atmosphere of reduced oxygen and increased carbon dioxide. The selective media must contain blood or charcoal to remove toxic oxygen radicals and antibiotics to inhibit growth of another microorganism. Campylobacters are slow-growing organisms, usually requiring incubation for 2 days and longer. Campylobacter fetus is not thermophilic and can grow in 48 degrees. However, its isolation requires a microaerophilic atmosphere. Here you can see the growth of Campylobacter on the blood agar. Small, smoothie, uncolored colonies. Main antigens is O antigen and flagella H antigen. Virulence factor, flagellum to invade, protein adhesins, cytolethal distending toxin, lipo-oligosaccharide, and enterotoxin. These factors are involved in several pathogenicity-like properties that can be divided into bacterial chemotaxis, motility, attachment, invasion, survival, cellular transmigration, and spread to deeper tissue. All of these steps require intimate interaction between bacteria and host cell, including immune cell, enabled by the collection of bacteria and host factors that have already been identified. Epidemiology. Campylobacter infection are zoonotic, with a variety of animals serving as reservoirs. Human acquired the infection with Campylobacter jejuni and Campylobacter coli after consumption of contaminated food, milk, or water, especially with contaminated chicken and poultry. Campylobacter infection is in developed countries are very spreaded, and also Campylobacter absaliensis may be acquired with domestic dogs, with contact with after contact with domestic dogs. Food products that neutralize gastric acid, for example, milk, effectively reduce the infection dose. The main route of transmission is fecal oral, and from person to person also may occur, but it is common for the disease to be transmitted by food handlers. This is most commonly observed in infants and young children. Pathogenesis and immunity. Campylobacter gastroenteritis is typically a self-limited infection managed by the replacement of lost fluids and electrolytes. The risk of disease is influenced by the infection dose. The organisms are killed when exposed to gastric acid, so conditions that decrease of neutralized gastric acid secretion favor disease. Campylobacter jejuni disease produces histologic damage to the mucosal surface of jejunum, ileum, and colon. The mucosal surface appears ulcerate, edematous, and bloody with script, abscesses, and the epithelial glands and infiltration of lamina propria with neutrophils, mononuclear cells, and eosinophils. Another immune-related late complication of Campylobacter infection is reactive arthritis, a condition characterized by joint pain and the swelling involving the hands, ankles, and knees, and persisting from 1 week to several months. Immune-related complication is because of causing autoimmune activity by Campylobacter. Campylobacter jejunum and Campylobacter coli rarely cause bacteremia. However, Campylobacter fetus has propensity to spread from the gastrointestinal tract to the blood and the distal foci. Bacteremia is particularly common and debilitated and immune compromised patients, such as those with liver disease, diabetes, chronic alcoholism, and asthma. Here you can see interaction. So bacteria goes to the lumen and mucus layer, and then interact with epithelium and attachment and invade to the cell, and then it can go to the macrophage and destroy macrophage, and in some cases it can go to the generalization to blood stream and cause bacteremia. But bacteremia is more often for Campylobacter fetus, not by jejuni. Laboratory diagnosis is by microscopy, antigen detection, ELISA, and nucleic acid-based tested PCR. Uh, cultivating is not performed because of difficulties, and it is more easy to detect Campilobacter by PCR or by ELISA, um, and that's why cultivating method, pure culture method, is not applied now. Treatment with antibiotic, erythromycin or azithromycin are antibiotics of choice. Also, tetracycline and fluoroquinolone used as secondary antibiotics. Systemic infection are treated with aminoglycoside, chloramphenicol, and imipenem. Uh, Exposure to enteric Campylobacter is prevented by proper food preparation, particularly poultry, avoidance of unpasteurized dairy products, and implementation of safeguard to prevent contamination of water supplies. That's all. Thank you for attention. And if you have any question, please ask me in WhatsApp.